Good morning, class 9 students. This is your biology teacher, Agrima Rawat. I hope everybody is healthy and doing their studies regularly. Okay, I expect everybody have seen the part 1 of this chapter 5. Okay, now we can continue with the part 2. That in the part 1 we have talked about the introduction of the cell, in which we have talked about the discovery of the cell, cell theory, cell size, cell shape. Now we are going to talk about the components of the cell, by which the cell has been composed of. So you can see in this picture that there are many components of the cell. Okay, so we are going to talk about them one by one. The first thing we are going to talk about is the plasma membrane. Plasma membrane is also termed as a cell membrane. It is the outermost covering of the cell that separates the content of the cell from its external environment. Now you can understand it from here. From this diagram, this blue lining, that is the plasma membrane. Plasma membrane is also termed as cell membrane, right? So you can see it here, that this membrane is separating this inside of the cell components, and this is the external environment. So this is the separating this content of the cell from the external environment. This means it is going to protect it from the external environment, right? Now when many of the scientists started working on the structure of the cell, two scientists came forward, Singer and Nicholson. They gave the model of the plasma membrane structure that stated that plasma membrane is a continuous lipid bilayer. Continuous lipid. Lipid is a substance, a chemical substance. You can see it here, the circular structure and the two yellow legs. So this is one lipid, and this is a continuous layer of the lipid. Downside also you can see this lipid. This is the second layer of the lipid. That's why it is called as continuous lipid bilayer. Bilayer means two layers, right? So this is a continuous lipid bilayer having integrated proteins. The proteins, blue structure, this blue structure, this blue structure, these are the proteins. These are being integrated. You can see it here. In here also this protein is here. So this protein is being embedded inside the lipid layer. That's why it is called as integrated protein molecules. These lipids and protein help out in the transportation of the material throughout from inside to outside of the plasma membrane. And other than the protein and lipid, cholesterol, polysaccharides are also there that help to give the energy to the plasma membrane to do these all work. Okay. And this membrane is semi-fluid. Semi-fluid means that much flexibility that it is not that flowy as a water and not solid as a brick. So it is having a average flexibility like as a balloon. You would have seen if you have blown a balloon and you just press it out, so its structure takes a different shape. So that is the same flexibility as the plasma membrane. Okay, and dynamic means that a thing that can change its shapes according to the environment. So this is the whole thing, and the structure, that the model of the plasma membrane is being called as fluid mosaic model. Why it is called as fluid mosaic model? Fluid means flexibility. Okay, mosaic means the structure that is made up of different things. Different things, like you can see it here, lipids are here, proteins are here, polysaccharides are here. So all of them are making this structure. So that's why this structure's name have been given as fluid mosaic model. Understood? Okay, now this plasma membrane is also termed as selectively permeable membrane. So selectively permeable membrane, why? Because it selectively permit few of the things to come inside or go outside the cell. Okay, so few of the things are allowed to go inside and few of the things are not allowed to go inside the cell. It is like a border security force. of our country that do not let everybody to come inside the country or go outside the country, right? So this membrane protect us from many harmful things. Okay, now we are going to talk about that how this in and out of the material or substance takes place. This takes place by a process called osmosis. But before going to the osmosis, we are going to talk about few terms that are that has to be known by use to understand osmosis. Solvent, solute. So solvent and solute, when we are talking about that, we can understand by a solution. Okay, solution is something that has been two substances are been mixed to each other, a solvent and solute. So this solution is of sugar water, or you can say a sugar syrup is here. So water is the substance on which the other thing is going to be dissolved. Okay, and the sugar is the thing that is going to be dissolved inside the water. So the liquid in which a solute is dissolved to form a solution is called as solvent, and solute is the substance we dissolve into a liquid to form a solution. Are you getting my point? So the water is the solvent and the sugar is the solute. Other than sugar, there can be anything else as a solute, and other than water, there can be anything else in the place of the water, right? So the solvent can be of many types, but we are taking the example of sugar water here. So the water is the solvent and sugar is the solute. Okay, now we are going to talk about the concentration. What do you understand by concentration? So taking an example for it. Okay, so this is the example. We have taken two jars. In the jar 1 and jar 2, in both we have taken same amount of water, 100 ml of water in both. In the jar 1 we have taken only 10 gram of sugar, and in the jar 2 we have taken 50 gram of sugar. Okay, now we have made a solution from it. Now when we are talking about the concentration of the solution, in which jar do you think the concentration of solution is higher? Very good. The jar 2. Jar 2 have more solute amount, right? Because the solvent amount is same, but sugar is higher in the jar 2. So the jar 2 concentration of solution is higher and than the jar 1, right? Now understand this next question, that in which jar the concentration of solvent is higher. Are you getting my point? Concentration of solvent. So in both of the jar, the amount of water is same, but in which concentration of solvent is higher? We are going to compare it with the solute. So solute is less in the jar 1, and in the jar 2 the solute is higher. So in comparison, in the jar 1 the solvent concentration is higher than the jar 2. Are you getting my point? So this is what about the concentration is. Now as we know about the concentration, so we can understand the osmosis very easily. But with the osmosis we also have to take diffusion. Diffusion is one of the process, but we are going to talk about that also. So with this diagram we can easily understand this. So first of all, we should be knowing the difference between the osmosis and diffusion. In the diffusion we talk about the solute molecules movement from high to low concentration. Where the solute molecules is higher, it is going to move from higher to lower concentration. But in the osmosis the solvent molecules only move. Understand this very carefully. In the osmosis only solvent molecules are going to move. Okay, so you can see it here in the diffusion. This is the solvent particles and these are the solutes, the red particles. So in this particular area the solute amount is higher, and in this area the solute is not there. So the concentration is higher here and lower here. So the solute molecules are going to move from the higher to lower concentration. So they are going to diffuse throughout the solvent, right? So this is how the diffusion takes place. So in every place it is going to be equal. By the time it is not equal, the diffusion will keep taking place. As the equilibrium is being found, the diffusion process will stop. Now coming to the osmosis. So I have told you that solvent molecules only pass. But why? Because there is a semi-permeable membrane involved in this, in the osmosis process. Semi-permeable membrane is like a sieve, जैसे छन्नी होती है. छन्नी से सिर्फ आपकी लिक्विड चीज़ें जा सकती हैं, right? Solvent is a liquid, right? So the solvent only moves through the semi-permeable membrane. Okay, so if we are going to talk about the two sides of this beaker, in this side, on the left side, you can see it here that the water on the both sides, the solvent are same, solvent amount is same, but the solute is lesser on the left side and solute on the right side is higher. Okay, so when we are talking about the concentration of the solvent, that is higher on the very good left side it is higher, the solvent amount is higher, concentration of solvent is higher. Okay, so from left side to the right side, the movement of the solvent will take place. So this is here, the solvent from the left side has gone to the right side to equalize the concentration of the solution. Are you getting my point? So this is how the osmosis takes place. So one of the difference between diffusion and osmosis is in diffusion solute molecules move from higher to lower concentration, but in the osmosis solvent molecules move from the higher to lower solution. Okay, do not go through this definition. It is also the same, but what I am telling you, just understand that the osmosis is the movement of solvent from the higher to lower concentration. Are you getting my point? Okay, and semi-permeable membrane is not involved in the diffusion, whereas in the osmosis the semi-permeable membrane is involved. Okay, now we are going to talk about the direction of osmosis. From if the osmosis will be it will be taking from inside to outside or outside to inside. It depends on the solution in which we have put down the cell. So this yellow thing is the cell and this is the external environment of the cell. Okay, so if the cell has been put in a isotonic solution, in the isotonic solution means the concentration inside the cell and outside the cell are same, right? So there will be no net water flow. Net water flow means if 10 ml of water came out of the cell, then 10 ml of water will go inside the cell. There will be equal movement if the movement is taking place. So in the isotonic solution there will be no net flow of water. Iso means same, so isotonic solution means same concentration inside and outside the water, but just understand this. Then no water flow will be there. When we are talking about the hypertonic solution, so in the hypertonic solution the concentration of water inside the cell is higher. Are you getting my point? The concentration of solvent is higher inside the cell and concentration of solvent is lower here. That's why from the higher concentration of solvent to the lower concentration of solvent, the osmosis will take place. Okay, now you will get confused with this, that this is the hypertonic solution. Solution should be higher in something. So higher in tonic means solute. Okay, that's mean in the hypertonic solution, in the solution means the outer surface, surrounding of the cell, the solute particles are going to be higher. That's mean the solvent is automatically lesser. Okay, so that's whole thing is here, that this the solvent from the inside the cell will come out of the cell, and in this the cell will shrink matlab सिकुड़ना. Okay, so let us take an example for it. You would have seen raisins means kishmish. The raisins are being made by the grapes. So the grapes are being put in the sugar solution, high concentrated sugar solution, in which a large amount of sugar is there. So in comparison, the grape is having more solvent than the sugar solution. Then the water from the grape, inside the grape, will come outside of the grape, okay, in the solution. So the grape will start shrinking up. Okay, you would have seen the raisins, they are shrinked, right? They are the shrinked grapes. Okay, now coming to the hypotonic solution. I have told you hypo means less, tonic means solute. The less solute is here, so that's mean the solvent is automatically higher outside. In the solution, the solvent is higher, so the movement of the solvent will be from the outside to inside. Okay, are you getting my point? In this, you can take an example of any type of dal or rajma. When you put rajma inside the water, your mother soaked them in at night, so you would have seen the amount of rajma is less. But when you see it in the morning, that the level of the rajma goes up. Why? Because the rajma have taken the water inside it. That's mean the water you have the rajma has been put in the hypotonic solution. That's why the water have gone inside it. Okay, so the water was higher outside and inside it was lower. That's why the water have gone inside. Okay, so this is how it is. Now there are two types of osmosis. First is endoosmosis. When the cell is placed in the hypotonic solution, the water will flow into the cell. We have seen it here. You have put the cell in the hypotonic solution, then the water is going inside the cell. This type of osmosis is going to be called as, very good, endoosmosis, when the movement of water is inside. Endo means inside. Okay, now exoosmosis. Exo means outside, exit, out, bahar jane ka rasta, right? Exoosmosis, when a cell is placed in hypertonic solution, water flows out of the cell. You have seen it here, that when the cell has been put in the hypertonic solution, the water was going outside the cell, right? So this type of osmosis is called as, very good, exoosmosis. Okay, now we are going to talk about the endocytosis. What endocytosis is? Endo means inside. Cyto word is being called for the cell. Okay, so endocytosis is the process of ingestion. Ingestion means intake of the food or anything. The ingestion of material by a cell through the plasma membrane. So in here we are taking an example of amoeba. You have talked about it, that amoeba takes its food, this is the food of amoeba, by a structure called pseudopodia. The pseudopodia has been made by the plasma membrane, by the expansion of the plasma membrane, and it's take the material inside its body. The ingestion process is taking place, right? So this process of intaking the food by the amoeba is called as endocytosis. Are you getting my point? Endo means inside the cell. The ingestion has been taken place. So this process is known as endocytosis. Okay, so this was it for the plasma membrane. I hope you would have all understood it. In the next part, we are going to talk about the other components of the cell. Thank you.